Hello, my name is Dr. Dean Betts. I'm an associate professor in the Department of Physiology and Pharmacology at Western University in London, Ontario, Canada. So the title of this project is The Extra-Telomeric Functions of Telomerase Isoforms. And so this proposal focuses on the telomerase reverse transcriptase or TERT subunit, which is the catalytic subunit of the enzyme telomerase, which is well known to synthesize telomeric repeats, so the end structures of our chromosomes, the caps of our chromosomes, to provide chromosomal stability and in cells that have expressed high levels of this enzyme can confer their cellular immortality or prolonged proliferative capacity. So in stem cells, in highly proliferative cell types, this enzyme helps maintain a long proliferative potential. And obviously in most cancer cells, this enzyme is re-expressed and allows cellular immortality of probably over 80, 85, 90% of most cancers because of its telomere stability properties. However, over a number of years that my group and much research done by a lot of other groups have shown that telomerase has extra-telomeric roles. And I won't get into all of them, but one of the most recent roles that have been attributed to telomerase, extra-telomeric roles, and this is work primarily done by Steve Artandi's group in Stanford recently, shown that telomerase can bind as a transcriptional cofactor and modulate gene expression, in particular in the Wnt beta-catenin pathway. And so extra-telomeric telomerase can modulate gene transcription and affect stem cell function, which is very exciting. The focus of this proposal, however, is on the alternative splice variants of TERT. And so some of these, and there's many that are now known, but these have been known for a number of years. And for, I guess, the past 20 years, there's been limited studies on these, and most of them have basically attributed the roles of these alternative splice variants of TERT to be basically vehicles to modulate telomerase activity levels. Some of the deletion variants, and there's a few of them named alpha, beta, gamma. These are deletions of exons in the reverse transcriptase domain of the TERT pre-mRNA molecule. And so those deletion variants don't have the ability to synthesize telomeric repeats, and so they don't have the ability to, they don't have telomerase activity, and they don't have the ability to maintain telomere length. It's surprising that these alternative splice variants are expressed in various tissues during development, but also, and in particular stem cells and progenitor cells, and also various cancer and tumor cell lines. Cell types that typically maintain telomere length and a prolonged proliferative capacity. So with the recent work by Steve Artandi's group and others showing that full length or constitutively spliced TERT that synthesize telomeres can have this extra telomeric role in modulating gene transcription, it is quite possible that these alternative splice variants of TERT too as well have these extra telomeric roles in modulating gene transcription. So that's the aim of this proposal, is to one, do these alternative splice variants have this extra telomeric role, and two, can we identify novel transcriptional, binding sites for these alternative splice variants and the constitutively spliced TERT isoform as well, and also identify any novel protein-protein interactions that may help regulate TERT and alternative splice variants of TERT in regulating this gene transcription in stem cells, But in the future, we hope to look at the alternative functions of these alternative splice variants in various cancer cell lines as well. So this I Am a Scientist proposal is looking for seed money to help my group accumulate some more preliminary data, in particular doing some chromatin immunoprecipitation chip sequencing or chip-on-chip studies to identify some of these novel promoter or DNA binding regions that these alternative splice variants might bind to regulate gene transcription, and also some mass spectrometry studies to help identify any novel protein-protein interactions that TERT and some of these alternative splice variants of TERT may have to help regulate this gene transcription in stem cells. So that's the synopsis. I'm really excited about this project. I am interested and happy to collaborate with scientists from anywhere. I do so right now and obtain a number of clones of particular splice variants to carry out some of these studies. So if you're interested in learning more, please contact me, and thank you. Bye-bye.